With the Research, a webinar series brought to you by the GeneConvene Global Collaborative. Glad you could join us. My name is Dave O'Brockton. I, along with my colleague Hector Kamada, will be hosting and moderating this series. Before our speaker, Dr.Andrea Crisanti, begins this seminar, I'd like to tell you a little bit about GeneConvene Global Collaborative and make a few remarks about what's coming up in this webinar series. The GeneConvene Global Collaborative is an initiative within the Foundation for the National Institutes of Health. It's arisen from and really is an extension of the FNIH's long-time involvement in the vector-borne disease space. The potential of genetic biocontrol and gene drive technology specifically to contribute to the reduction and even the elimination of human malaria, for example, as well as other urgent public health challenges, is substantial, although with considerable uncertainty. The responsible exploration of these technologies will enable their efficacy, safety, and sustainability to be evaluated. The GeneConvene Global Collaborative works to advance the safe, ethical, and rigorous exploration of gene drive and other genetic biocontrol technologies. It does this by trying to anticipate emerging issues and to facilitate the development of guidance and best practices through consensus building. GeneConvene also seeks to offer accurate information, advice, and technical support for the purposes of fostering responsible approaches to research and governance of gene drive and other genetic biocontrol technologies for public health. This webinar series is a part of that effort. I encourage you to learn more about the GeneConvene Global Collaborative by visiting fnih.org/GeneConvene. GeneConvene has also created the GeneConvene Virtual Institute, which is a knowledge resource that aggregates, tracks, organizes, and shares the latest gene drive from scholarship, research, to media reports, to policy and regulatory documents. The Virtual Institute is striving to create a resource where those interested in gene drive and genetic biocontrol can come and learn about these classes of technologies. To help those interested in these areas of research and development to keep up with the accelerating activity in this space, the Virtual Institute publishes a weekly newsletter that notifies you of and points you to new scholarship, media reports, etc., related to gene drive and genetic biocontrol technologies. Check it out, and I also encourage you to follow us on Facebook and Twitter. I'd like to point out that the Virtual Institute is looking for a web assistant. This is a remote work contractor position that will require web-specific knowledge as well as some knowledge of biology. And fundamentally, the position is to provide support in developing and maintaining the GeneConvene Virtual Institute's digital presence. You can find out more about this position availability at the website below. Let me just say something very briefly about the webinar series that you're partaking in today. It's been a series of 6 webinars. We're in the 5th of the 6. These webinars have focused on the technical aspects of gene drive and really bringing in some of the leaders in the field of gene drive research to talk about their research in very technical terms, really to other scientists, and to provide them with an opportunity to get feedback from their colleagues. Following this webinar series, we'll have another series along the theme of regulatory and policy considerations. And here we have a series of 5 webinars scheduled that will cover a range of aspects of the policy and regulatory space. So we encourage you to keep track of what's happening here at the GeneConvene Global Collaborative, and you can register for these webinars. We'll give you that registration information soon as we get it organized. Let me just briefly take care of some housekeeping activities here before we get started. If for any reason you have technical issues, you can contact Tara and Deetria, 2 of our FNIH event staff, at the email address that's shown, events@fnih.org, and they'll be able to monitor that email and help you with any technical issues that you may be having. We're looking at a presentation that will go roughly about 50 minutes. Most certainly encourage you to ask questions and to feel free to use the chat function to do that. Also, would encourage you to do that at any time during the presentation that you have a question that comes to mind. We'll handle all those questions at the end of the presentation, however, and we'll do that again by using the chat function that you can find on your Zoom platform. We'll end after 90 minutes, and if there's any remaining questions, we'll address those offline, and the presentation is going to be recorded. The posted recording will be on the GeneConvene Virtual Institute at the web address shown below. Great, I now have the pleasure of introducing today's speaker, Dr.Andrea Crisanti from Imperial College in London. Andrea is a professor of molecular parasitology in the Department of Life Sciences at Imperial. He's also the director of the Center of Functional Genomics at the University of Perugia. Andrea is a leader in the field of vector biology, and as you hear today, he is a source of great technical innovation when it comes to the development of gene drive systems as tools for malaria elimination. Andrea was also responsible for the creation of a one-of-a-kind research facility in Perugia, Italy, that enables the testing of gene drive-containing mosquitoes in large flight cages that allow the insects to display behaviors of great importance in the field, such as swarming. This unique experimental system is providing insights into genetically modified mosquitoes that could not have been discerned under standard laboratory conditions, and is proving to be a really valuable asset. I also want to mention that Andrea played a central leadership role in developing a European-wide initiative to create research capacity for the implementation of genetic control of mosquitoes called InfraVec, and it's now on its second iteration called InfraVec 2. Finally, and I couldn't let this opportunity to go by without mentioning this, that Andrea has been playing a major role in Italy's response to COVID-19, becoming popularly known in the press as the father of swabs. Andrea's work on COVID-19 has been recognized widely, and some of you in the U.S. may have seen him on a popular TV news magazine show called 60 Minutes. So good morning from Washington, Andrea, or should I say Father Andrea, and good afternoon to you in Italy. Thanks for joining us. Thanks for joining us at the end of your day. Are you about ready to go?
Yes, I'm ready to go now.
Okay, let me turn it over to you. Tell me when you have your presentation up, and I'll confirm.
Yeah, this is now I think the presentation is on. Can you see it?
Yes, I can put it in presentation mode, and I can hear you, and you're ready to go. Thanks a lot for joining us.
Lovely. Thank you very much for a very generous words. It's a great opportunity for me to reconnect with the scientific community working on gene drive and vector control. I've been asked to provide information about gene drive and the state of research, and particularly this will give me the opportunity to touch on several aspects of the work we are doing in our laboratory. Just a few slides on the issue of malaria, because we apply gene drive in the field of vector control, particularly as a tool to eliminate malaria. At the moment, there are about 220 million people are infected every year, and there are about 400,000 deaths, mainly children, and malaria accounts approximately for 10% of our childhood deaths every year in Africa. Is the fourth highest cause of death. Well, the malaria control has improved over the years. Between the year 1990 and 2000, we've seen an increase in malaria cases throughout the world, particularly in Africa. And then after, let's say, a coordinated effort, there's seen a dramatic increase in funding in different vector control measures. We've seen a steady decline of malaria cases for at least 2 decades, but now since 3 years, malaria case have remained constant, and we don't know how this pandemic will affect the level of funding and the effort to control malaria, as well as other diseases that affect resource-poor countries. We will see probably the effect of this crisis maybe in the coming years, and highlight again, if necessary, the need to develop additional measure. Well, just to remind everybody that malaria in Africa is transmitted by 4 mosquito species: Anopheles gambiae, Anopheles colucci, Anopheles arabiensis, and Anopheles funesto. Our main goal is to develop a control measure against these first 3, Anopheles gambiae, Anopheles colucci, Anopheles arabiensis. Well, the reason why we don't work, or we haven't focused too much on Anopheles funestus so far, is because it's still very difficult to breed, and gene transfer technology are still being developed. So this is what I will talk today. Available vector, I will review briefly available vector control measure, and I will highlight why some of them works. I will elaborate a little bit on the rationale for developing GM mosquito and gene drive. Then I will talk about validating gene drive solution for Anopheles mosquito. I will talk about our development of gene drive targeting female fertility gene and how they work. Then I will go on the problem of resistance, how this can be overcome. And then I will talk a little bit about development of gene drive neutralizing technology. I will start now on malaria control measure targeting the vector, have proven to be the most effective. If you do not have mosquito, you don't have malaria transmission. So in principle, this is a very simple statement. And as you can see that there is a different measure targeting mosquito have a different effectiveness. And I would say the most effective is treated mosquito nets. Now, the reason why they are the most effective is because they're affordable, they are easy to implement, and they are sustainable. In principle, you don't need much effort to distribute bed nets, to explain how they work, and in principle, they are kind of culturally neutral, so they are accepted by the population. And the fact that they are so affordable, the fact that they are easy to use, in principle, that accounts for their success. And so I think the major message that we take from this is that a vector control measure, in order to be effective, has to be adopted by large by the population, has to be cost effective, and has to be sustainable. So this means that the particular country must be able to afford that particular measure. Now, if we now extrapolate the use of current intervention, and this is a document from the WHO Global Technical Strategy meeting, what it tells us, it shows the situation in 2016, and it shows the situation in 2030 with the current level of funding. So that means that with the available control measure and available funding, we won't be able to eradicate malaria by 2030. And this, again, additional question now arise by this epidemic that also is causing, it will cause some serious economic drawbacks. Now, a most important thing to remember is that the ability to transmit malaria by mosquito is genetically determined, and then therefore genetic modification could potentially impair the vector capacity. So in principle, this concept summarize our rationale for developing transgenic mosquito. And the transmission phenotype is influenced by longevity, feeding behavior, support of parasite development, reproductive rate. These are very complex phenotype, but they are genetic determined. So the concept is, if you can genetically manipulate mosquito, you could theoretically interfere with the ability of mosquito to transmit malaria. For example, you could develop genetic modification that shorten the lifespan of mosquito. We know that only mosquito they are older are able to transmit malaria, because of course they have to to live longer the parasite life cycle, have to feed at least twice. Feeding behavior, you could manipulate the ability of mosquito to feed on human rather than animals. You could interfere with critical molecule that support parasite development in the mosquito, or eventually you could reduce the ability of the mosquito to reproduce and therefore reduce the number and so the ability to transmit the disease. And this slide shows one of the first transgenic mosquito that we developed. This is a genetic modified larva that expressed the GFP on the dorsal ganglia. Now, the challenge here is, well, you can theoretically develop any such a mosquito. There are now, thanks to the genome sequencing, a number of very interesting candidate genes that eventually could allow you to reach any of the objective that I have listed before. However, the challenge is to spread the genetic modification from few individuals into a large population. You could, for example, develop a mosquito resistant to a parasite, reduce fertility, maybe even make it susceptible to insecticides or reprogram the behavior. However, the major obstacle to do this are the law of heredity. Now, mosquito, like all sexually reproducing organisms, they receive a set of chromosomes from the father and a set of chromosomes from the mother. So they have a 50% chance to inherit a particular trait, but they also have a 50% chance to transmit this trait to the follow generation. The outcome of this is that a frequency of particular trait doesn't change in a population unless a selection force is acting on this particular trait. So this means that if we release 30% of genetic modified mosquito, after several generations, we'll end up with 30% and so on. Now, mathematical model tell us that in order, for example, to have a particular effect on vector competence, for example, releasing mosquito that do not transmit malaria, you have to reach about 19%. So this means that you need to release 10 times as many mosquito as those that you find in the environment, and this is clearly is not possible. So, and this in principle highlight the need to overcome the low heredity of transmission of genes. And there are several natural examples that are able to do so and overcome the Mendelian inheritance, and they're called gene drive. They are transposable element, gamete killer, driving sex chromosome, maternal segregation distorted midea, and homing endonuclease genes. I remember very vividly about 15 years ago when my friend and colleague Austin Borg came to my office and highlight the potential of homing nucleases to act as gene drive, provided they could be engineered to target Anopheles genes. And so we start to collaborate and working on this field using endonucleases as a tool for developing a gene drive able to overcome the low heredity and spread in a target population a gene that could potentially interfere with malaria transmission. The mechanism of gene drive is very simple. They are endonucleases, so in principle they are genes that when transcribed, there are enzymes that cut the DNA at particular recognition sites. Now, the good thing about homing nucleases is they have a very, very long recognition sequence. So this means that it is very rare in the genome. Possibly they cut the genome only one time. Now, if the recognition site is interrupted by the sequence of the endonucleases, when this endonuclease is activated during the process of meiosis, they induce a cut in the intact chromosome at the site of recognition, and then you trigger the repair mechanism of the cell that in gamete is mainly by homologous recombination. So in principle, then the sequence of the nucleases is copied by repair on exactly the cut site. In this way, the genetic modification is transmitted to all offspring. Then I must say that we have tried homing endonucleases, and we succeed in developing a functional gene drive using homing endonucleases. However, we were not able to reprogram them to target Anopheles gambiae genes. So in order to show that they were functioning, we had to introduce the target site within the Anopheles gambiae genome, and at the same site we introduced the homing endonuclease that we show that it could spread. We also have tested other endonucleases like zinc finger and talon meganucleases, and we showed that they also can drive. So we came with the concept that any nuclease can be engineered to work as a gene drive, provided that is activated at the right time and at the right place. The right time is during meiosis, and the right place is in the germ cell. Now, gene drive can be used, can be adapted to two basic strategies to fight malaria. One is population suppression, releasing modified mosquito into the population that in principle have the ability to interfere with the fertility of either the male or the female. And then over time, the net effect is that you reduce the size of the population. Or they can be used to replace the population. So for example, you link the gene drive to a genetic modification that, for example, is able to block malaria transmission, interfere with the ability of the parasite to develop in the mosquito. Then after a few generations, then you spread the trait in the population, then the population can become refractory. In principle, the two are not mutually exclusive, and actually there are some theoretical studies that shows that maybe they can be used sequentially together to have an enhanced effect. Now we have focused on a gene drive approach for population suppression. One is based on female sterility. This means to spread in a population a genetic modification that would impair female fertility. And the other one is to engineer a genetic modification that would distort the sex ratio toward males. This has the additional benefit that males do not transmit malaria. And will progressively replace the population with male up to the point that you do not make any more male to female fertile couple, and then the population crashes. Now, this is a slide that shows the molecular component of a CRISPR-based gene drive. So in principle, what you need to make a gene drive is very simple. So in principle, you need a reporter gene, you need a germline promoter, you need the nuclease, a Cas9, and then you need a guide RNA. And you need to engineer this particular construct at the site that you want to, in principle, to cut. Now, we have used CRISPR nucleases because of its flexibility. So in principle, we adapted immediately a CRISPR technology, and we attempted to target 3 genes that we identified in the Anopheles gambiae genome that probably were involved, according to the data from Drosophila, in female fertility, which are noodle, yellow G, chitin binding. And these slides show, for example, the phenotype of heterozygous mosquito female that have been engineered to destroy 1 copy of this gene. Of course, this approach will work only if the gene is not insufficient. So this means that if you have 1 copy destroyed, the mosquito is still fertile. In fact, you see here that all these 3 genes, in principle, satisfy this condition. If you destroy 1 copy, for example, this chitin bind, you still have high fertility. If you destroy 2 copies, in principle, you have no egg laying. So in principle, you destroy fertility. So in principle, this is the larvae deposited by individual mosquito. I should say that this experiment has been done by not by introducing the gene drive, but by simply destroying the gene of interest. Now, when you do the gene drive, then that is able to modify the inheritance pattern, And in fact, you see that the majority of the progeny have inherited the construct. This is the chitin binding protein, both male and female, the yellow G and the noodle. So in principle, the gene driving is able to work and to distort the inheritance pattern. And non-homologous end joining are detected at a very low frequency. Now, unlike the data that we observed when generating heterozygous and homozygous mosquito by CRISPR-mediated editing, when we generated homozygous mosquito by gene drive, we found something which was totally unexpected. We saw that the heterozygous mosquito were fertile, whereas in the case of the male, whereas the heterozygous female were not fertile. Well, the reason for this is that during the process of gamete formation, you have an excess of production of CRISP, which is deposited in the egg and in principle destroy the other copy when the sperm cell fertilizes the egg. So in principle, these are mosquito that in a way they have a kind of chimeric, because somatic they are heterozygous, but in the germline they are homozygous because the deposited Cas line has destroyed the allele inherited by the father. Anyhow, for some case, for example, for this one, there was enough fertility to justify a gene drive experiment that we carried out. And in principle, what we did, we seed the wild type mosquito population with a gene drive mosquito. As you can see, this is the predicted spread of the drive, and this is the to replicate that. This is what we observed at generation 7. The drive has reached about 75%. Then we continued the experiment for other generation, and we observed that in both cages, the frequency of the drive dramatically decreased. And then you can see here in red the predicted dynamics of spread, and in black the experimental points. So we thought that this was due to the emergence of resistance. And now there are many ways how resistance can emerge. This is the target site. This could be that there is a variant allele that in principle are not susceptible to the cut, to the nuclease activity. So in principle, generation after generation, they spread, and in principle, they counteract the effect of the gene drive. However, the gene drive activity of the nuclease can generate resistance. For example, here we have the nuclease that induce a double strand break. Now, this can be repaired by homology direct repair, and in principle, this is not a problem because this mosquito will still have a drive that will work. Or you may have non-homologous end join, and this can invariably result in a mutation that modify the sequence and then the ability of the endonucleases to cut the target site. And then this will have, if this mutation generates a functional version of the gene, this has a tremendous selective advantage in the population. And this is exactly what we observe. Now, this is the slide, a different way to represent the slide that you've already seen. The blue and the orange are the two cages. These are different level of frequency of the CRISPR allele in the population at different generations, and you see that they decrease progressively. However, at different points, what we did, we collected mosquito and we individually sequence. We did pooled sequencing at different time points, and then we analyzed by deep sequencing. And what you see here in black is the wild type mosquito. In blue, you see the mutation that is induced by the gene drive. So in principle, this is a gene drive mutation, and what you see here is a resistant allele. And you see generation by generation, the frequency of mutant allele increase in frequency up to the point that at generation 20 you find or you select only mutant allele that are only in frame, and so they are not cleaved by the nucleases, but they encode for a functional protein. No, this is sorry, is the same slide. Now, there are several strategies where you can mitigate the rise of resistance to gene drive. One is the target site choice. So in principle, one possibility is to choose a target site that cannot be mutated without having a functional impact on the encoded gene. Then you can eventually restrict nuclease expression so that it is only expressed during the time of meiosis. Minimize the occurrence of resistance. Now, this is a very valuable source. It's the Anopheles Gambia Genome Project databases, and this we use to search for conserved target candidates that, if destroyed, would impair female fertility. And also we search for sequences that were conserved not only within the Anopheles gambia genome, but also in all other mosquito species that are related to Anopheles gambia. And we found a sequence which is very interesting, which is a sequence which is at the boundary between intron 4 and exon 5 of double sex. As you can see, this particular sequence is extremely conserved in Anopheles gambia, Coluzzi, Merus, Arabiense, Quadrenulatus, and Meles, but is also very conserved in other species. And it shows some divergence only in very distant species like Arthroparvus and Albimanus. So what we did, we engineer a gene drive that would cut this sequence here and the intron exon 5 boundary. Just a few words about double sex. Double sex is a critical genes that control the differentiation program, alternative differentiation program into male and female mosquito. As you can see, the only difference between male and female is that female must have exon 5, whereas in male the genomic sequence is spliced in such a way that the exon 5 is skipped. In fact, this is the male transcript, as you can see, miss this particular region. So we destroy exon 5 of double sex in male and female. And now what you can see here is a phenotype of an Anopheles gambia male, XY, in which both copy of double sex have been destroyed, and this, believe me, looks like a normal male. This is an heterozygous female, and again, this looks very much as a normal female. However, when in the female you destroy both copy of double sex, in principle, you start to see some features that we call intersex. For example, the mouth parts here are not those typical of a female, but resemble those of a male. You start to see this plumose antenna, and then you see this male clasp, which are dorsally inverted, which are the typical of male, which are external reproductive organ of the male. Now, when you look at the internal organ, you see that in principle these intersex, they have accessory gland, which are here, they don't have ovaries, and you can see some you call it, yeah, you can see some very primordial structure that resemble testicle. Then we develop a gene drive targeting double sex at the intron 4 exon 5 boundary, and we observe consistently population crash. Now these are 4 independent experiments, and in orange, in blue, red, and green line. And we see that in all cases, in a matter of 8 to 14 generation, all cages crashed. Now, this is in gray, you see stochastic iteration of the model that takes into account some factors like, for example, the fitness as reproduction and so on. And as you can see, as you increase the frequency of the transgene, you dramatically decrease the egg output up to the point that no viable mosquito are hatched. Then we also looked at the frequency of mutation at the target site, and we observed a number of mutations. So this particular construct is not immune to generate a mutation by non-homologous end join event. However, none of these mutations, as you can see here, there are deletion substitution. None of these mutations increase in frequency in time. So this means that all of these mutations are likely to lead to an unfunctional splice event or to an unfunctional exon 5 sequence. We, given these very promising results, we also test this construct in a large case study. These are, as David mentioned to you, this is a facility that allows us to study mosquito under conditions that mimic more closely those that we find in malaria endemic area, like in terms of temperature, humidity, and as well as low intensity and wavelength from dawn to sunset. And indeed, in these mosquito, in these cages, in this environment, the mosquito show behaviors that are more similar to those that we find in the wild in terms of survival. So in the small cages in the lab, the mosquito can survive up to 20, 30 days. In the large case, we see that the female and male survivor is close as what is anticipated to be in the field, which means anything between 10 to 15 days. But most important, they also have a completely different mating behavior. In the large case, the mosquito swarm. So we set an experiment, a replicate experiment, in which we tested, of course, the control, and then we released the gene drive at 25 and 50% frequency. I should also not omit to say that in this experiment, we could also set up a system where we also study the effect of overlapping generation. And in principle, this is the data that we collected at 12.5 allelic frequency, at 25 allelic frequency. And as you can see, this is the egg output, and as you see that the egg output decrease as you see the increase of the gene drive, up to the point that after about 300 days, in one case for 12.5% allelic release, and about 250 days, in principle, the population crashes. Now here you see a lot of oscillation here, and this is due to the particular setup of the experiment and to the overlapping generation. Then we also attempt to develop a gene drive in which the double sex has been targeted to different highly conserved sequence. One is here. We already discussed this at the boundary between intron 4 and exon 5, but there is also another highly conserved sequence here at the 3' hand of the exon 5. So in principle, we developed a gene drive in which a guide was designed to cut here and another one to cut here. And in principle, we observed that this particular solution provide equal or even better transmission bias. Here you can see that we have, in principle, anything between 99 to 100% transmission bias, irrespectively whether the gene drive was in the female or in the male parents. And also the fertility is extremely good. Now, we are still in the process of testing this in gene drive experiments, and as well as in large cages. So in principle, I do not have yet the data for this, and for sure, the COVID epidemic is partially responsible for this delay. As I mentioned to you, another approach to achieve population collapse is to develop a sex ratio distortion for vector control. We were very lucky because one of the homing endonucleases, IPPO1, is able to cut a highly conserved sequence in the ribosome, and ribosome sequences in mosquitoes, Anopheles, gambiae, all cluster in one chromosomal location. So in principle, this shows the data that you get in terms of male progeny, so and in terms of hatching rate. And in principle, sex distortion theoretically is wide applicable across insect species. So in principle, here you see that the male progeny increased dramatically. Here is a wild type. This is what you get when you introduce this particular endonuclease that cuts the ribosomes in a very highly conserved sequence, but you can also use CRISP to get the same result. And now there is one thing to say, is that this particular genetic modification was not possible to put on the Y, because as you will see in a moment, in principle, this is particularly effective if you're able to engineer this particular modification on the Y chromosome, because what you expect is the genetic modification is on the Y chromosome, destroy the X, and you inherited only Y-bearing gametes that have the genetic modification. So this is the rationale. Unfortunately, this is not possible, because during meiosis, what happens? You have meiotic sex chromosome inactivation. So the Y and the X chromosome are very early condensed and inactivated. And so in principle, it's not possible to express any gene at the right time, at the right place. So in principle, if you put nucleases, is designed to destroy the X chromosome. During meiosis, in principle, you do not get expression, because the entire chromosome is silenced. And this is a phenomenon called meiotic sex chromosome inactivation. It's an epigenetic silent process that occurs exclusively in germ cells. And we have tried different attempts to overcome this process, but we have not been successful in expressing sex distorter on the Y. There are several, however, what we did, sorry, I think they are mixed up here. What we did, we developed a construct targeting the double sex locus at the boundary between intron 4 and exon 5. And this construct also not only was able to cut the double sex, but also carried a gene that was able to destroy the X chromosome. In particular, it carried the IPPO gene. And we call this a sex distorted gene drive. And in principle here, what you see is the fraction of transgenic male, and this is in the case of the female, so this is the inheritance and this is the fraction of male. So as you can see, when you use this construct here, in principle, this is a construct, I should say, this is of course only active in male. So you see a dramatic bias in the progeny towards male, and you see that they are all transgenic. In female, of course, you should not expect to see any bias because the construct is only active in male, but you see, nevertheless, a bias in the formation, in the production of a female. So it's a very high oming, 97% in male and 99% in female, and there's a very strong male bias. So in principle, you get 94% of the male of the progeny are males. And when we did invasion and suppression cage population, we in principle observed that the construct spreads very, very fast. We had 2 cage at 10% allelic frequency and 2 cage at 50% allelic frequency. You can see that in 5 generations, the cage at 50% allelic frequency are completely 100% male and transgenic. Male is blue, is blue, and transgenic is dashed. So you see they immediately reach the fixation high frequency of male. While the construct increase in the population, you have a parallel and dramatic decrease of egg laying, and so practically the population crashes. We also, as I mentioned to you, trying to develop a system that could counteract the CRISP. This mainly for regulatory reason and also to understand how this system could be modulated. And there are some bacteria, there are some genes that are able, that mainly found in prophages, that are able to counteract the action of CRISPR. And 2 of them found in Listeria monocytogenes, which are called ICR2, ICR24, they're able to block Streptococcus pyogenes Cas9 when tested in E. coli and human cells. So in principle, we tested this anti-CRISPR and to see whether we could block the gene drive. We were in the fortunate condition to have a working gene drive, the double-sex gene drive, and so we developed a model to test the activity of this inhibitory protein. Now, first of all, before testing the activity of this anti-CRISPR gene or construct in a gene drive population, we tested whether they were able to block a gene drive in crosses. So in principle, this is a gene drive, and this is an anti-drive. They are cross. You select the double heterozygous, so you cross with wild type, you have a different combination, and in principle, you expect that in principle, you expect 50% of the inheritance of the anti-drive, and if the anti-drive works, of course, you expect about 50% of the drive to be inherited. Of course, if it doesn't work, of course, the anti-drive would exceed the 50% inheritance bias. And now this is what you obtain. So in principle, if there is only the anti-drive only, you expect 90% of the anti-drive of the drive. If you have the anti-drive only, you expect 50%. And in principle, the experiment shows that you are able to block the anti-drive. So the drive. So this means that the anti-drive is able to block the inheritance of the drive. And so we move to make a gene drive experiment. So in principle, these are the result of the experiment setup that I showed you before. This is gene drive plus anti-drive, and you see that in principle the gene drive usually is inherited at the frequency of 50%. So this shows that the anti-drive is working. If in the same experiment you put the gene drive only, you see that it behaves normally giving an inheritance bias ranging from 95 to 100%. This is the inheritance pattern of the antidrive only. So the antidrive only is inherited at 50%, as you expect. The gene drive is inherited according to what we've seen before, between 95 to 100%. If you put them together, you see that the antidrive is inherited at 50%, as you would expect, but also the gene drive is completely neutralized. So in principle, you bring back the gene drive with a Mendelian inheritance pattern. Then on the basis of this observation, we set up an experiment to assess whether the antidrive was able to block the spread of the double-sex gene drive. So these are two cages. This is the gene drive, cage 3 and cage 4, and this is the spread of the drive. It's in principle is the same construct that we have seen before. Is the double-sex gene drive, as you can see, is spread very quickly. In 10 generations, they both reach 100%, and in parallel, you decrease the number of wild type and the number of egg, and both cages collapsed after one after 10 generations, another one after 14 generations, although this is not, at the time we did this slide, this is not completed. Now, this is the same experiment when you release 50% of gene drive and 20% of antidrive. As you can see, over time, the antidrive frequency increases, and the gene drive is not able to collapse the population. I should say here that unfortunately we have seen a fitness effect of the antidrive, and this is the reason why the antidrive does not increase to the point that it reaches 100%. We model this, and if this antidrive did not have any fitness cost, it would have completely counteract the effect of the gene drive. And I think I stop here. And I want to thank my lab here in London on the left panel, and on the right panel, the lab internally that is responsible for all the activities of the large cages. And thank you very much for your attention.
Great. Thank you, Andrea. Thanks for a great talk. And I'd like to encourage our listeners to chat in their questions, and at any time, and we'll begin to take those as they come in. So we do have a question. Andrea, I'm going to read this question to you, and you can respond. This is a question from Mike Smansky. Is Cas9 activity in mosquito population, is Cas9 activity in mosquitoes temperature dependent across the range of temperatures the drive might experience in the field?
We have tested Cas9. Now we have tested Cas9 activity both in vitro and in vivo, and the answer is that yes, is temperature dependent. However, in the temperature we breed mosquito, which is 26 degrees, we don't see a dramatic decrease over 37.
Okay. Great. So again, I'd like to encourage people to ask questions. I do have one that I'll ask while people are preparing their questions. Could you give us your thoughts on which of the strategies that you've been developing you think might be most suited for programmatic use?
I think that the double-sex gene drive is very robust. We have now, I think, more than 12 replicas of this gene drive experiment. We've never seen a single case of resistance. Now, the gene drive, sex and the sex distorter are also very promising. We have a faster dynamic of spread, And also, I did not have here the time to discuss, but also we model also the occurrence of resistance by the fact that you decrease then progressively the number of female, you also decrease the probability of developing resistance, because most of the resistance, of course, occurs in female. This is where the selection pressure occurs. And of course, there are a lot of regulatory issues about the release of these mosquitoes. One of the reasons why we developed the anti-drive to show that we also have them available to block the spread of the gene drive.
Mm-hmm. Great. We do have a question from one of the audience. Let's see. Let me get that question up here. It's from yes. So the question is, I hope I haven't missed this detail, but what should we expect to happen if we make regular introgression of wild type mosquitoes in cage experiments with suppression gene drives?
This is a very good,
this is a very good.
Yeah.
Would it have a little effect?
Yeah, no, it will have an effect. Yes. Now, if you release a wild type mosquito on a gene drive, you dramatically counteract the effect of the gene drive, and if you release massive amounts, you probably block the gene drive all along.
Right. Okay. Here's another question. Again, this is from Mike Smansky. What is the limit of detection for resistance mutations in your large cage experiments? So, i.e., what is the total number of offspring in a normal experiment?
Well, it can be quite big, because usually this kind of we have calculated that for each experiment, you in principle generate about 300,000 eggs. Of course, you don't test the eggs; you test only the adults. So in principle, you test a sample of this population. Now we run some modeling to check what is the probability that we have missed some resistant allele. And unfortunately, I don't remember the answer to this question. Because, I think we did, and I think it was anyhow. It was kind of low. I don't remember now. I should remember the occurrence per generation, but and the probability that we missed it, and I don't remember this data very well.
Okay, great, thanks. I'm going to go on to another question. This is from Moaz Khan. He says, Moaz says, thanks for a great talk. In the data you showed, the inheritance of the anti-drive seems to be slightly skewed above 50%. Don't know if that's statistically significant, but might there be a reason for that? Just thought it was curious.
Well, the anti-drive has the homozygous status as a fitness cost. So we haven't tested that the homozygous status. It could be that this is the reason.
Mm-hmm. Okay. Thanks. We have another question here from one of our listeners, one of our attendees, Pratima Chanuri. With the double sex gene drive, do we see only non-functional resistant alleles as the egg output decreases dramatically and might actually lose the functional resistant alleles?
Sorry, I didn't get it.
Yeah, I'm gonna say it again. And I'm with the double sex gene drive, do we see only non-functional resistant alleles as the
Yeah.
egg output decreases dramatically?
Yes.
And yeah, and might you actually lose the functional resistant alleles?
I should also say, I mean, I didn't have the time. I didn't have the time to present here, but we developed a genetic system to carry out a saturation mutagenesis of intron 4 exon 5 sequence. I think we have generated a total of several thousands of events. Not a single one generated a functional allele.
Mhm. Okay.
Going
Let's
back
move
to
on to another question.
sorry, going back to the
Yeah,
previous
go ahead.
going back to the previous question, are for the skew of the anti-drive. Well, as I mentioned to you, the anti-drive at the heterozygous status as an important fitness. Now, at the heterozygous status, yes, there is a slightly increase in inheritance, but I don't think it's statistically significant. So.
Okay. Here's a question from the next question is from Max Scott. Max says, "Great talk. Is the semi-sterility of double-sex drive females also seen with the two guide RNA lines, RNA lines?
Yes.
Does the level of semi-sterility vary between lines? Is that genetic background?"
Yeah. Actually, I must say that we unfortunately the COVID pandemic has slowed down a lot our experiment, but this double guide line is performing extremely well in terms of fitness, in terms of fertility, in terms of bias, inheritance bias. So yes, we didn't see, and also in terms of fitness, so we didn't see any fitness costs.
Okay. Here's a question from Ava Sirninath-Sinji. She says, "Thank you very much for the talk. Can I ask about the phased approach to the target malaria releases? With the repeated application for phase 1 releases in Burkina Faso being submitted, what is the reason to repeat this trial? And will data be published soon on the 2019 release?"
Actually, I must say that I was not involved in the implementation in Burkina Faso, so I might have incomplete knowledge of what has happened. I don't have what has been done by Frederic Trippet. We obtained permission. We released the line in July. I can tell you that they were kind of promising in terms of survival, in terms of ability to mate with wild type mosquito, as well as we started the spread, the range of spread. Now we are filing a new release, and with a modified construct, actually.
Okay. Let's see. At the moment, we don't have any other questions. I'm going to ask a question here just to see if there's give people time to further ask questions. Andrea, could you say something more about the facility that you described for the large cage studies? It was unique, and I wonder if you might spend a few minutes describing that facility and what makes it unique and how it's different from some of the lab cage experiments, and how and if you've actually been able to see things in those experiments that you would never have been able to see in the lab.
Yes. For example, okay, let me start with the facility itself. I mean, it consists of several large which can be partitioned in smaller cubicle. Let's say typically they are about 15 by they are about, let's say, anything between, they are different size, but let's say they are about anything between 100 to 200 cubic meter. So in principle, they're pretty big. They have quite a complex system to control the temperature as well the humidity in order to make sure that you don't build a gradient. They have quite sophisticated that you can reproduce faithfully both the intensity and the weight down sunset, and actually you can also change temperature and humidity and light according to what you record in a region in the field. Now, when you put mosquito in this cage, you immediately, the most obvious things that you see is that the lifespan is reduced. Rather than a lifespan of 35 to 45 days, typically in a small cage where in the lab, we have a closer to what we see in the field. So in the field, we exert a selection pressure on the use of energy. They also have a feeding behavior. The one that we observe in the field, they start to swarm around some particular food that you can put on the pavement of the cage. We usually start to see the male, and then you see that the female go toward the couple. And we also did this experiment with the double sex gene drive, which has and we observed some dynamics which escape attention if we have carried out in small cages. For example, we have seen that if you release mosquito, for example, only once, and then you do the second release 10 days. So in principle, you have a very, very low of the two release, and this may create a lot of problem in the dynamic of a spread. So in principle, we learned that if we want to release mosquito, we can't just rely on let's say, to release a part of 3 or 4 days, but ideally we should make several releases, in order to avoid this huge frequency, which may eventually lead to one generation. So something that was somehow unexpected, or at least we didn't think when we experimented. And we also
great. Yeah, go ahead.
For example, on the sorry, I have a very fastidious noise. I will try. Maybe it's because your microphone is on, David. If you switch your microphone, maybe it's better for me. Okay.
Actually, I'm going to mute it now. It's been muted.
Yeah. Okay. Yeah, it's much better now. If we also since you can change the temperature in this cage to reproduce exactly what happened in the field from day to day, and within the same day you can reproduce the 9-day excursion. We were able also to test stability of particularly construct, and we have some particular solution are not suitable for the field.
Okay, great, thanks. I want to back up to a question that was asked previously. I must say that your audio is sort of degraded a little bit with the talk. It was great during the talk. It's breaking up a little bit now. I don't know if that's anything you have control over, but I'm just mentioning it. And I want to go back to the question that was asked by Ava about the releases in Burkina Faso. Some of your audio broke up there, and we had a little trouble hearing some of your response. So I want to repeat the question, and let's give it another shot. So Ava says, again, thank you very much for the talk. And can I ask about the phased approach to the target malaria releases with a repeated application for phase 1 releases in Burkina Faso being submitted? What is the reason to repeat this trial? And will data be published soon on the 2019 release? So,
okay,
thanks.
Now, the 2019 release was a success. We were able to release a target number of mosquito, collection experiment to test the spread of the mosquito to assess whether they had made it. We now another application, but this is a different genetic. The initial application was a sterile line. Now, the second one is a sex distorter line, which utilize the same IPPO gene under a different which is modified, less stable, and so able to overcome the problem of the sterility. So in principle, it lines in prepared time and generates male. So there is not two identical applications.
Right. So her follow-up question is: so there's no repeat of phase 1.
No, I mean it's let's say which has a higher complexity of construct. The first one was sterile mosquito, which expressed the IPPO1, but the IPPO1 since was carried over by the sperm, was active in the female and killed also the female X chromosome. So this line was there. Now we developed the variants of IPPO which are less stable, and not carry over by the sperm. So now this line is fertile. But of course, it's not a gene drive because the distorter is not on the Y.
Right. Thanks. And I'll just do another follow-up from Ava. Do you know when phase 2 will be released? Well,
no, we don't know yet. I mean, we had a timeline that we have been dramatically affected by the COVID. So at the moment, some experiments are on hold, and some revised.
Great, thanks. Going to move on to another question here. We still have some time. This is from Andrew Roberts. He says, thanks for a really nice talk, covering a lot of information. This may be a bit of an outside question, but have you had any conversations with regulators about the anti-drive system? From a technology standpoint, it seems very feasible and probably effective, but it seems like it will be challenging to ask decision makers to get on board with an idea of releasing a second engineered construct as a mitigation method to control a previously released construct.
Okay, well, the answer is that the regulatory want to have a tool in hands if something goes wrong. So we had conversation. They said to us that they really are very, very interested to the development of an anti-drive, because the main concern is that we cannot the long term. So knowing that you can block the drug provides a lot of comfort. Of course, you need to make a different application for the anti-drive. We are perfectly aware of this. But I don't think it's resolved the issue when you apply for the gene drive, you also need separately to apply if something goes wrong. But we developed the anti-drive on the basis of the feedback that we got from the regulator. They really would like to have on it. We would counteract the effect of the drive if something.
Right. Thanks. Let's see here. Right. So I think we've come just about to the end of our questions. I don't see any more questions coming in. So maybe this is a good point to stop. And let me just say a few closing remarks. So again, Andrea, thank you for a great presentation. And I'd like to encourage everyone in the audience to join me in thanking Andrea for a wonderful presentation.
Thank you very much. Thanks a lot.
Great. So I'd like to say thank you to the attendees. Thank you for all the questions that came in. I'd like to remind you that the remaining webinar in this particular series of webinars will be next week at the same time and place. It will be given by Mike Smansky from the University of Minnesota, and he'll be talking about a very unique system that he's been developing called engineered genetic incompatibility. And I encourage you all to attend that webinar. So again, with that, I'd like to say thank you again to Andrea. Thank you to you for attending, and we'll see everybody next week.